Welcome to today's lecture on cell biology, where we will dive deep into the fascinating world of molecular mechanisms and their implications in various biological processes. In this session, we will explore a range of topics, including molecular chaperones, protein folding, post-translational modifications, PTMs, and their significance in health and disease. So let's jump right in. Molecular chaperones are specialized proteins that assist in protein folding and prevent misfolding. They work alongside co-chaperones, which fine-tune their activity. Heat shock protein 70, HSP70, is an example of a well-studied chaperone, aided by co-chaperones like HSP40, HSP90, and HSP110. This chaperone network ensures proper protein folding and maintains cellular homeostasis. Protein disulfide isomerase, PDI, is an enzyme in the endoplasmic reticulum that helps proteins fold correctly by catalyzing the formation and rearrangement of disulfide bonds. PDI works with other proteins like ERP57 and ERP72 to ensure proper disulfide bond formation, which is crucial for protein function. Disruption of this process can lead to protein misfolding and cellular stress. Despite the intracellular machinery's vigilance, protein misfolding and aggregation occasionally occur, leading to the formation of insoluble protein aggregates. These aggregates can disrupt cellular processes, cause cellular toxicity, and give rise to various disorders, including neurodegenerative diseases. Prion diseases, such as Creutzfeldt-Jakob disease and Alzheimer's disease, involve the accumulation of misfolded proteins with a beta sheet-rich conformation. These misfolded proteins can convert normally folded proteins into the pathological form, propagating the disease within the brain. Understanding the mechanisms underlying protein aggregation and the unique properties of prions is crucial for developing therapeutic strategies aimed at preventing or reversing these devastating diseases. Intrinsically disordered proteins, IDPs, defy conventional understanding of protein folding, highlighting their functional importance in cellular processes. IDPs have flexible structures that undergo transitions upon interaction, enabling adaptability. Exploring their folding mechanisms has enhanced our comprehension of protein function and potential therapeutic opportunities. Multimeric protein complexes are intricate assemblies composed of multiple subunits, contributing distinct functional units. Proper assembly and stability of these complexes are critical for their functional integrity. Post-translational modifications, PTMs, play a crucial role in the assembly and regulation of multimeric protein complexes. PTMs such as phosphorylation, acetylation, and methylation can modulate subunit interactions, stability, enzymatic activity, and affinity for other cellular factors. These modifications dynamically regulate complex formation, turnover, and response to cellular signals. Comprehending the intricate interplay between PTMs and the assembly of multimeric protein complexes provides insights into how these complexes function and how their dysregulation can lead to pathological conditions. Summary: Sumoylation is a covalent attachment of small ubiquitin-like modifier, SUMO, proteins to target proteins. This process, involving different enzymes, affects protein localization, stability, interactions, and function, influencing various cellular processes such as DNA repair, transcriptional regulation, and responses to stress. Understanding sumoylation enhances our understanding of cell regulation and may lead to therapeutic applications. And glycosylation is an essential process that adds carbohydrate chains to proteins. It occurs in the ER and is important for protein folding, stability, and trafficking. The ER also has quality control systems to ensure only properly folded and glycosylated proteins leave. Misfolded proteins are degraded in the cytoplasm to maintain a healthy cell. Histone modifications are post-translational changes to histone proteins that regulate gene expression, DNA repair, and chromosome structure. These modifications include acetylation, methylation, phosphorylation, ubiquitination, and sumoylation. They act as marks on chromatin, recruiting proteins that either activate or silence gene expression. Epigenetic regulation mediated by histone modifications is important in development, cellular identity, and disease. Dysregulation of these modifications can lead to abnormal gene expression patterns and contribute to diseases like cancer. Understanding the complexity and dynamics of histone modifications yields insights into epigenetic regulation and potential therapeutic interventions. Protein phosphorylation plays a critical role in cell signaling, affecting protein function, localization, and interactions. By adding phosphate groups to target proteins, protein kinases modify their properties and activities. These phosphorylation events are crucial for signal transduction cascades, allowing information to be relayed from cell surface receptors to the nucleus. Abnormal protein phosphorylation is associated with diseases like cancer and neurodegeneration, making targeted therapies that restore proper phosphorylation states a potential treatment approach. Studying the intricate dynamics of protein phosphorylation provides deeper insights into cellular signaling networks and potential avenues for therapeutic interventions. Ubiquitin-mediated proteolysis is a crucial process for maintaining protein balance and turnover in cells. Ubiquitin gets attached to target proteins, marking them for degradation by the proteasome. This can happen through mono or polyubiquitination. The proteasome recognizes polyubiquitinated proteins and degrades them into small peptides. Proper protein turnover is essential for normal cell function, and dysregulation of this process can contribute to various diseases. Protein folding diseases, also known as conformational diseases, arise from protein misfolding and the subsequent aggregation of abnormal proteins. These diseases include various neurodegenerative disorders, such as Alzheimer's, Parkinson's, and Huntington's disease. Mutations or alterations in protein structure can render proteins prone to misfolding, leading to gain or loss of function, toxicity, and cellular dysfunction. Misfolded proteins can form oligomers and aggregates that disrupt cellular processes, impair proteostasis, and contribute to disease progression. Unraveling the molecular mechanisms underlying protein folding diseases is crucial for developing therapeutic strategies to prevent or alleviate their devastating effects. Post-translational modifications play a central role in cancer biology, contributing to tumor initiation, progression, and response to therapy. Aberrant PTMs can alter protein function, localization, stability, and interactions, directly impacting oncogenic pathways. Molecular players involved in PTMs, such as kinases, phosphatases, acetyltransferases, and deacetylases, are frequently dysregulated in cancer. Consequently, PTMs are often exploited as targets for cancer diagnosis, prognosis, and therapy. Understanding the intricate interplay between PTMs and cancer biology provides critical insights into disease mechanisms and paves the way for the development of targeted therapies. In conclusion, protein folding and post-translational modifications play critical roles in cellular function. Their dysregulation can lead to diseases such as cancer. Further research and exploration in this field will advance our understanding and pave the way for new therapeutic interventions.